afternoon, ladies and gentlemen. So I'm very happy that I had a nice introduction also already done by Dominique. So I can focus on the things that I know best, which is more the scientific part. So as a scientist, I will show you actually the lab processes. Actually, just one thing, when we say biorefinery scientist, actually it means laboratory scientist. So we have a special word to say laboratory for biorefinery. But this here, so I will concentrate it on the workflows and especially for the DNA extractions. There I will give actually 3 examples, and where we will really see the workflow that we do in IBBL for DNA extraction. And first of all, it will be from blood or from Buffy code. Then I will give an example, well, how we automated this and what's the outcome together with the method validation data. And also from low volumes of Buffy code, where we extract also DNA. And also from another sample type, which is actually a feces, so stool samples. And for all of these, I will show some data that we have produced in IBBL, and which I would like to share with you today, in the sense of automation, of course. So just a small word, so this will only be the only introductory slide. We collect, after the informed consent, so human body fluids, especially for the fluids for urine, blood, saliva, and many different ones. We have automated the liquid handling part. And for this, for example, as Dominique already mentioned, we use a Tecan Evo 200 Freedom liquid handler. And I will also show one of the applications in a small video afterwards. Also, one important biobanking activity is that we routinely do also extractions of RNA and DNA, so of nucleic acids, from our samples. And for this, we use 2 different platforms. Well, we separate the instruments to extract RNA and DNA so that there can be no cross-contamination. For RNA, so it's the BioRobot Universal and QIAcube that we have in-house. I will not have time today to concentrate on this one, maybe in the next presentation, but today I will talk mainly about the DNA extraction, and there we use the chemagic MSM I, which is actually a magnetic beads-based technology to extract DNA, and it's also coming with a liquid handler, which is the Janus liquid handler. So the first example. So this is the plasma and Buffy code preparation for later on DNA extraction. And for those of you actually who are not maybe so familiar with this, first of all, we collect blood in EDTA tubes. Then it's shipped at room temperature. And when we centrifuge the tubes at 2000 G for 10 minutes, so this is our standard protocol, then actually we get this kind of sample here. But here already the plasma was pipetted, so there's only a small volume left. But so in orange here you have the plasma, here below you have the red blood cells, and the intermediate layer is the one which interests us particularly for this presentation here today, because this is the Buffy code. So it's a small little white layer, and which contains all the cells, as mainly white blood cells, with the nuclei, and so you can extract the DNA. So just before I come to the DNA extraction, some words about the plasma aliquoting. So what we, as a standard protocol, prepare is 12 aliquots of 220 microliters plasma. And this is automated on the Tecan liquid handler. And we could show in a validation study that these plasma samples actually are fit for purpose, and mainly for proteomics and immunological parameters testing. So proteomics has already quite high, stringent levels of quality requirements, and so we could show that our samples are fit for this purpose. So for the Buffy code preparation, we also didn't want to do this manually, so this was also automated on the Tecan liquid handler, and there we'll show you the video on the next slide. And then afterwards we did the DNA extraction on the MSM I extractor, and we could show that this Buffy code is fit for purpose for genetic, epigenetic, and also genomic analysis. And I will give details on the next video. So what's the challenge actually of automating this Buffy code extraction? First of all, for automation you want to do several samples. Of course you don't have always the same volume into your blood tubes. So first of all, you will have differences in the volumes because, for example, in this script that we use, we can automate 24 samples at the same time. But then also there are individual variations, even for same volume of initial blood volume, where you have difference in the buffy coat composition. So you might have a big buffy coat, you might have a different texture. It might be more or less viscous from one patient to the other one. This makes it really a challenge to automate this. And what we, and together with the field specialists, came up with was to pipette this buffy coat very slowly in spiral movements. And here I will show you the video. Hopefully it will work. Okay. Well, that's maybe a little bit too much noise because it's not so noisy as this. Well, okay, here the sample actually, oops, that stopped the video. Sorry. Just wanted to show you. Doesn't want anymore? No? Another try. Okay, so it will pick up the sample. It will show it in front of a barcode scanner. The barcode, the 2D barcode is identified, so sample identification. Now each sample goes into this unit, which is a tube inspection unit. There's actually a laser which measures at which level is the buffy coat. This information is then transmitted to the pipetting unit, which will exactly go to the position where is the buffy coat for that sample. So really individual. So here you can see the liquid handling arm, which picks up a tip. And now it will exactly go into the buffy coat layer and do this spiral movement. You will see it a little bit. There will be a small zoom, but it's very slow. So actually this is something which can be done for 8 samples at the same time. And operators are, well, it's difficult to do this manually, as you can imagine, aspirate so slowly. And we could show that this was really nicely done. And well, that's the video. So it takes a little bit of time, but I think it's now almost over the video. Yes. So you see the spiral movement. Yep. Okay, that is part of the video. And then afterwards, it's put in a tube where we do then DNA extraction. So when this aliquoting is done on the TCAN of the buffy coat, afterwards we extracted the DNA. And all of the extractions were done on the MSM I, so which is actually the magnetic separation module from Perkin-Elmer, always in the same conditions as for the same conditions for all of the samples. And what we could show when you compare automatically prepared buffy coat compared to manually prepared buffy coat, actually for the first script that was done on the TCAN, we had quite the same amounts of extracted DNA, around 150 micrograms. But when we then optimized the script, actually here we could reach around 200 micrograms of DNA. So this was even more than when the buffy coat is prepared manually. So what we can say as a conclusion here was that and it was coming from 364 donors, that there's a significant increase in DNA quantity obtained with the optimized buffy coat isolation script. These data, together with other data which I don't have time to show today, were also published in 2012 in the Biopreservation and Biobanking journal. In the meantime, actually, this extraction was semi-automatic. In the meantime, we upgraded our system and we integrated a liquid handling platform, which is the Janus, and which you see actually here as the white part. So all of this is the liquid handling part, and the actual extractor is this blue part, dark blue part over here, together with the arm. And what will it help us to automate this part? So what does the Janus help us? Especially it is to do the extraction run preparation, so the liquid handling of the buffers, to add also the magnetic beads, and to pipette the samples. Afterwards, so then it goes into this machine, the extraction is done, that part fully automated. When it comes out again, it's in a rack. So you still need to transfer your DNA ELRIGs into tubes. So also this, the Janus can do. So it will transfer the DNA ELRIGs into storage tubes. And what we also integrated is a quantification run, so that a plate is prepared, which you can put immediately on a spectrophotometer, and you can read the sample quality parameters like DNA concentration and DNA purity. So you have immediately the quality data of the samples, which are also automatically prepared just after the DNA extraction run. So this completes the whole workflow from the centrifugation of the samples, the pipetting, the buffy coat preparation, the extraction, and the preparation of the plate for DNA quantification. So just to tell you some words, for the liquid handler, there is also the 2D barcode scanner here, which in the beginning also we had, of course, some integration problems, but finally now it works really well, because these are all, our labels are 2D barcodes. And also one part of the Janus liquid handler is actually a 2D bottom barcode scanner, so that in our storage tubes, they all have a 2D bottom barcode. And so on this instrument here, you can scan the whole rack, and you have the sample identification of the 96 samples that you prepared. And here is actually the space for the preparation of the quantification plate, and the space for the tips that the liquid handling arm can pick up the tips from the platform. Okay, so this was for the first example. The workflow example 2, it's almost the same, but it's a smaller amount of buffy coat which is prepared. And this was in the sense of being able to process 96 samples at the same time, because the previous workflow, it was for 24 extractions at the same time. Here it's for 96. So here you see the platform where actually the different racks are positioned, and with the liquid handling arm will dispense then afterwards the buffers and transport these plates onto the rack, which then goes into the extractor. Here you see the 96 rod head, so which permits to do the 96 extractions at the same time. So also what we have automated here again, we have a different script on the TCAN, where you, again, from 8 milliliters of blood in the beginning, you extract 1 milliliter of buffy coat, but we only use 100 microliters for DNA extractions. The remaining 900 microliters are stored for other purposes or later on extractions, whatever we decide. But for this one here, we have a low volume, because also what we found out and what we see from the requirement of our clients, we don't need the big amount of DNA. The people want good quality, but nowadays NGS sequencing, you don't need a lot of DNA. And still here we get, out of these 100 microliters, we get approximately 14, 15 micrograms on average of DNA, and this is largely sufficient for the application we are asked for. So, okay, this I mentioned already, that Janus is able to transfer the L weights from the tubes into the storage tubes, and that's, so this is another workflow where we have low volumes and 96 rod head. So let's now move on to another sample type, which is the stool sample. And there, again, we did a method optimization and validation, but not only for the extraction method, but also for the sample collection tube. Again, these results were published earlier this year in the Biopreservation and Biobanking journal. So here you see the different collection tubes. Why is it important to check for different collection tubes? Because actually only 2 of these are plain tubes, so not containing any stabilizers or anything. But the other 4 here are containing stabilizing reagents, because as you can imagine, stool collection is most easy for the people to do it at home and to bring it the next day to the clinics or to the hospital. However, you may say, yes, but everyone has a freezer already. Not everyone wants to put this kind of samples in their freezer. But also there is the constraint that you might freeze it at home, and you bring it there, and in the meantime there is a small gap of time where it might start thawing. And this thawing is really critical for the quality of sample, especially of stool samples. And so we wanted to evaluate here the 4 different stabilizing containing containers compared to the plain tubes. So we had 6 different tubes, and we tested also 3 different extraction methods. what I forgot to say is that we had 3 donors, and for all of the 3 donors, we had 3 replicates for the 6 different container types and for the 3 different extraction methods. So we had more than 120 samples in total to test. And what were the analytical endpoints that we tested? We tested DNA purity, spectrophotometry for DNA yield, either for total bacterial or human DNA, but also fluorometry and also quantitative qPCR. Also, we tested for PCR inhibition assessment and metagenomics analysis of the 16S rRNA gene sequencing. And so today I will just have the time to show you these results here. So here is actually a representation of the relative abundance of the taxa present in the stool samples that we tested for donor 1, donor 2, and donor 3. As you can see, there is quite a big difference in colors, meaning that different bacterial strains and species are represented in these samples. This is what you can expect. However, what was more interesting to us, or surprising, is that when we had the same samples but for different extraction methods, there was also a difference. So actually the sample itself afterwards, what you find, the bacterial species which are present, depend on the extraction method that you choose. And what you can see is actually that the magnetic beads-based extraction method, the MSM I, is almost the same, giving, well, more closer, these 2 are more closer one to the other one, compared to the PowerMac. Well, the MSM I and the PowerMac are closer one to the other one, because they are both magnetic beads-based, compared to the all-in-one extraction kit, which is actually column-based, so silica membrane-based extraction kit. So this brings us to the conclusion that really the extraction method is a very important factor. And in-house, actually, we selected the method which gave us the best quality results. And so as a summary for this study here, we could say that the MSM I, so this is the instrument what I have shown quite often now until now, really had good quality samples. However, it was combined with this plain tube, which is actually called the brown tube, but this is a detail. This is a plain tube. It was not possible to do home collection. And so in this study, we concluded that from the samples which all had stabilizers, the container types which had stabilizers inside, actually the red one combined again with the MSM I extraction method gave the best results, because you can see a plus in all of the different parameters, either for the quality, but also for logistical parameters such as possibility of home collection and automation. And so what we can conclude is that the MSM I extraction method is fit for purpose. And as I told you, details, you can find it in the publication. So just one word. Yeah, I still have 2 minutes. It's one of the last slides. So for the DNA extraction from stool in IBBL, our standard operating procedure is now that we use the MSM I, 24 samples per run. It approximately takes 1.5 hours to run. And from 200 milligrams of fresh or frozen stool samples, we get in average around 9 micrograms. And for the moment, we are working on a solution also to integrate the liquid handling part into this. But it's quite tricky because stool samples can have different consistencies in the beginning. But we are working on it, and we hope to be able to come up with a solution very soon. So this brings us now to the last slide, which is my last slide, which is a little bit of the conclusions. It's a little bit, well, I put advantages and disadvantages, because as we have seen the different workflows, it was more illustrations. So actually from my point of view, from the lab point of view, what you can clearly say, advantages, that you have a high throughput, but only if you have standardized workflows. So if someone comes to you and asks for some special workflow, and you first have to develop a script, and you first have to validate that it works, and it gives the same results as a manual workflow, then the high throughput will take a while to be installed. So I put it only for the high standard highest throughput for standard workflows. But once it's installed, it will liberate time for the lab people to do other stuff, to do what they also need to do, but which is not the routine processing. And also for the quality of the sample, I think it's a very important aspect that the pipetting is really very precise and that you don't have this interoperator variability, which is quite known in the field of quality, that it is a main issue also. And then it's possible to do electronical tracking with the limb together combined with the limbs. Of course, disadvantage is you can always have the case that an instrument breaks down. Either you have a second one which you can immediately replace, like for us we have a second TCAN where we can put the same process at the same, continue the process at the same step on a different machine, or you need to find a manual solution. So at that moment also manpower is needed. For the disadvantages also, sometimes it's complicated to integrate instrument in the limbs. It depends also on the supplier, how should I say, motivation and availability to integrate these things together with us. And if you have, for example, one day where you only have 2 samples and you have a 96 well extraction plate, then your efficiency is not really high. But also for this, so the efficiency decreases if there's a low throughput. And then something that we underestimated in the beginning, but we now know it, that you really need to have in-depth knowledge of the software if you don't want to call each time the tech service who will tell you we will come soon. So that you can also do some changes, easy changes yourself into the scripts. And this actually asks quite a lot of time investment and knowledge also of the software. And this is for developing and testing new protocols. But what I would like to finish with is still that we are happy that we have the possibility to automate sample processes, and that it's a big challenge also, but it's a big chance also that we can do it. And with this, I would like to thank the team, especially the lab team. So Faye, who is our chief laboratory manager, and then also all the other people from the lab. And thank you all for your attention.
So, any question for Conny?
I was just wondering, when you do your DNA extractions, do you store that DNA in multiple tubes or just in a single tube?
We store it for the moment in one single tube because what we said also for to have a best efficiency of storage capacities also. In the beginning, we wanted to store it in 3 aliquots, but this was getting very difficult because you do small aliquots, and each time you do an aliquot, you have a little bit of volume loss. And so we decided to go for 1 aliquot, and normally we have elution volumes around 250 or 300 microliters, so it just fits into our 0.5 tubes. So we went for the 1 aliquot storage.
When a customer requests a sample, do you have to, is there a concern that you're taking a tube from a cold environment to head it to the customers and then freeze it back? Or is that
yes, but yeah, you're right. You will have a free store cycle, but actually for DNA, this is not a big problem. And we will also, normally what the customer says, they say, I want to have 1 microgram, or I want to have that many microliters. And so there will also, what we want to have is that we keep something also in the stock that for later on purposes. But yes, you will have a free store cycle, but for DNA, this is not critical. And also what's then possible that we do aliquots later on, because there might be samples which are really precious, and where we then decide, okay, now we do it aliquoting. And also, I should add also to this, so we do 1 aliquot for our routine sample collection, but if there is a special project where already the PI defines in the beginning, I need to have 5 aliquots, we will do it, although it's maybe not our standard process, but it depends on the project. Yeah.
Yes, just a question about the visual inspection to determine the buffer code zone. Do you also estimate the quantity to be pipetted by the TCAN, or how the TCAN stop to pipette?
Okay, so it will always pipette 940 microliters exactly, so approximately 1 tip of 1 ml. But actually, for us, it's not because we do DNA extraction afterwards. It's not important if it's they call it a dirty buffer coat, so meaning that it's contaminated with red blood cells. Actually, it's looking quite red afterwards. But for us, this is not important. So actually, it can just continue. If there is really a small amount of buffer coat, it can also pipette some white blood cells, as long as we have the white blood cells in there to do our extraction afterwards. Yeah.
Yes. Do you have any experience in extraction of stem cells from umbilical cord?
From umbilical cord?
Yeah. In some countries, I think it's forbidden, but I don't know in Luxembourg.
No, for the moment, actually, we don't do this. It's a very interesting field. It's, I think, for everyone. But for the moment, we are not doing this stem cells from cord blood. Yeah.
Other questions?
I was wondering, how do you validate an automated method? Do you just do a bridging between the manual and automated method, or do you test it? Do you test several validation parameters?
So for the method validation, a main part is really to compare, as you said, the manual against the automated part. But then also we have a continuous monitoring of our results. Like, for example, for DNA extractions, we continuously have the quantifications and the the DNA purity results. And so this we always plot it in graphs, and we see if there is a trend or something like this which decreases. But for the proper method validation, yes, it's mainly compare in several replicates, in several studies by different operators, the comparison manual against automated. Yes.
Thank you.
Thank you, Conny.
Thank you.